Hi everyone, I'm Hiruna Samarakon. I'm a genomic research software engineer at the Garvan Institute of Medical Research and I did my PhD at UNSW Sydney. Today I want to tell you about how we read DNA and a hidden part of that data that most people throw away. Let's start at the very beginning. Every living thing, including every one of us, carries a set of instructions and that is called DNA. Think of it as an instruction manual written in an alphabet of just four letters A, C, G, and T. There are about 3 billion of these letters packed into every single cell in our body. The letters also pair up in a fixed way A always with a T, C always with a G. If we want to understand disease or cancer or how life works, we first must read that manual. The technology for reading it is called DNA sequencing. One of the most exciting ways to read DNA is nanopore sequencing. The idea is simple. You take a tiny hole, a nanopore that sits in a membrane, and you pass an electric current through it. Then you thread a single strand of DNA through that hole. As it passes, it blocks the current by different amounts, and the current wobbles up and down. That wobbly trace is what we call the raw signal. Here's a way to picture it. Imagine a turnstile that everyone goes through one at a time. A runner, someone with a big suitcase, someone in a wheelchair. Each one passes through differently and takes a different amount of time. If the gate was sensitive enough, you could tell who went through just from how they pushed through it. A nanopore does exactly that with DNA. Each letter disturbs the current in its own way. Real DNA is messier than a queue of people, but the idea is the same. So what do people do with that wobbly signal? They run it through a software called base caller. Base calling software translates the wobbles back into the letters A, C, G, and T. And for most researchers, that's the end of the story. They keep the letters and they throw the raw signal away. Here's an analogy. It's like recording an interview, typing up the transcript and then deleting the audio. The transcript is useful, but you've lost the tone of the voice, the hesitations, everything that wasn't in the words. And it turns out the audio really matters. Hidden in that raw signal is information you simply cannot get from the letters alone. See that little M? That's a chemical tag on the DNA, called methylation. It switches genes on and off and is central to how cancer develops. The signal can also reveal DNA damage and the shape of RNA molecules. The letters alone are blind to all of this. But here's the catch. A single sequencing run produces terabytes of this raw signal, while the file of letters is tiny by comparison. So to save space and effort, most people just kept the small file of letters and threw the signal away. In fact, when we surveyed 100 nanopore analysis tools, only 12 made any use of the raw signal. Vast amounts of this valuable data were being discarded. And this is the gap my PhD set out to close. So we dug deeper and asked a simple question. Why were researchers throwing the signal away? It wasn't because they didn't know it was valuable. They did. They avoided it because working with the signal was genuinely painful. Think of the signal as a vehicle trying to travel down this road and hitting a speed bump at every single step. We mapped out the whole analysis pathway and we found 3 points where the signal hit friction. First, just handling the sheer volume of data. Second, reading the signal into DNA letters. And third, actually making sense of the signal once you have it. My PhD set out to flatten every one of these bumps to make using the signal a smooth ride instead of a painful one. Let me show you how we tackled each step. The first friction is sheer scale. A single sequencing run can produce terabytes of raw signal. Think of your phone's camera roll, but instead of few thousand photos, imagine millions of them, all dumped into one folder with no albums, no dates, no way to search. Everything you need is in there, but finding anything or even moving it all to a new phone becomes almost impossible. On top of that, the data came in the manufacturer's format, which was slow and awkward to work with. So I built a software called Slow5 Tools that fixes this. It converts the data into a faster, open format and lets you merge it, split it, and summarize it efficiently at the scale of hundreds of terabytes. And it caught on. Slow5 Tools has been downloaded over 7,000 times and installed more than 380,000 times. This work was published in the journal Genome Biology. The second friction is speed, turning the raw signal into letters. The step called base calling is heavy and it runs on powerful, expensive processors called GPUs, the same chips used for gaming and AI. You'd think the way to go faster is just to buy more of them, but when I looked closely, I found the real holdup wasn't the computing at all. The processors were sitting idle, waiting, because the data couldn't be fed to them fast enough. Picture a world-class chef who can cook anything in seconds, but the ingredients are being carried over one at a time from a storeroom across the building. The chef isn't the problem, the supply line is. So instead of buying more processors, I fixed the supply line. I changed how the data was stored and delivered so it could keep up. You can see the difference here. Each pair of bars is the same job, run three different ways, on a single computer, across many, and on the cloud. The tall blue bars are the old method, the short red bars are ours. Same work, a fraction of the time, up to 9 times faster with no extra hardware. And this work was published in the journal Bioinformatics. The third friction is the deepest one, and it really has two parts. Suppose you've cleared the first two hurdles. You can handle the data and you can read it quickly. You still hit a wall because the signal is just a long stream of numbers. And here's the catch. You can't really work with the signal until you can see it. If you can't look at it lined up against the DNA, you can't spot what's interesting. You can't compare one method against another. And you can't tell when something's gone wrong. But there was no good way to actually visualize these signals. So I built a software called Squigglizer that does exactly that. It turns the raw signal into an interactive graph you can explore, lined up against the DNA letters. For the first time, researchers could see their signal data properly. And it's now used extensively by labs around the world, including Professor Michael Schutz's group at Johns Hopkins. This work was published in the journal Bioinformatics. So what does being able to see the signal actually buy us? Here's the first example. This is a single spot in the genome where one DNA letter differs between molecules. Some reads carry an A here, others carry a T. That's a genetic variant, the kind of difference that can matter in disease. On the left, you are looking at it in the usual way, in the letters. On the right, you are seeing the very same spot in the raw signal. Notice that the two versions trace out clearly different shapes. The signal doesn't just agree with the letters, it shows you the difference directly in the physics of how each molecule moves through the pore. Being able to lay these side by side and actually see them is what makes this kind of comparison possible. But here's the part I find genuinely exciting. Remember those chemical tags? Methylation, that switch genes on and off, and that are invisible in the letters? Now watch what happens in the signal. This reads pileup has the same DNA letter C at position 38. In the letters, there is no way to tell them apart. Some of them are methylated while the others are not. And in the signal, you can see it. The signal traces are clearly different. This is something that was inaccessible before, but it is real, it's visible, and it's useful. Now, seeing the signal is one thing, but to interpret it, you need a kind of dictionary, technically a KMA model. On the right here is what it looks like, every possible short stretch of DNA listed alongside the current it produces. That's the lookup that tells you which wiggle in the signal means which letters. Without it, the signal is just shapes. With it, the shapes have a meaning. And this is where a real research need came in. A collaborating lab, Dr. Jonathan Guke's group at A-Star in Singapore, wanted to detect a specific chemical tag on RNA called M6A. But to do that, they needed this dictionary for their exact type of data, and it didn't exist. The manufacturer controlled it and was slow to release new versions, which left labs like they are stuck. And the dictionary is enormous. Its size grows explosively with length. The standard one lists every 9-letter stretch of DNA. That's over 250,000 entries. So I built a way to generate it from scratch, no manufacturer needed, and to shrink it dramatically down to every 5-letter stretch, just over 1,000 entries. That's more than 250 times smaller. And here's the key, it still works just as well. These curves on the left are the proof. My lightweight dictionary in color red sitting right on top of the big official one. Same accuracy at detecting that tag from a fraction of the size. And this work was published in Bioinformatics. So let me step back. Remember this road, a speed bump at every step? Here it is now. Piece by piece, my work flattened those bumps. Handling the data became smooth. Reading it into letters became fast. And the signal could finally be seen and made sense of. The signal now gets a clear run. And this isn't a thesis that just sits on a shelf. The tools are open source and being used right now at the Garvan Institute here in Sydney and at research centers across the UK, the United States, Canada, and Singapore. Together they've been downloaded and installed hundreds of thousands of times. And why does this matter beyond the technology? Because of the hidden information in that signal, the chemical tags, the modifications, is exactly the kind of signal that helps us understand human health and biology. By making the raw signal usable, we've opened up a layer of biology that was quite literally being thrown away. So it's time to bring it all together. We started with the simple problem. A rich layer of biological information, the raw signal, was being overlooked simply because it was too painful to use. People had turned a blind eye to it. My PhD built the tools to change that, to handle it, to read it, and to make sense of it, and to put those tools open and free into the hands of researchers around the world. And if there's one idea I would like to leave you with, it's this. Some of the most worthwhile problems aren't the ones nobody has thought of. They are the ones everyone has quietly turned a blind eye to, because they seem too hard or too tedious to bother with. The signal everyone overlooked turned out to be worth a very close look. I am deeply grateful to UNSW and the Garvan Institute of Medical Research for the opportunity to carry out this PhD research, and especially to my supervisors, Hasindu, Ira, and Sri. The software and papers are all freely available, and thank you very much for listening.